Hello and welcome back to today's lesson. Today we'll be focusing on 1.5, which is the origin of cells. And to begin with, we're going to focus on Louis Pasteur and his experiment. So first of all, we must state, and we already know that one of the rules of cell theory is that all cells come from pre-existing cells. Now, Louis, or Pasteur, gave an experiment to show how the cells come from pre-existing cells and disprove which was currently believed at the time of spontaneous generation. So he had 3 different flasks, all connected with a swan nose, which is what this thing is, how it dips down, kind of like a swan's neck. It's called a swan's neck glass, sorry. And so he left one as is. He broke the flask in one of them as well, and then he tilted one so that the liquid was coming out. So before each experiment, he boiled it so that any microbes that are currently in there would be dead, and in each flask was a solution of broth so that if there were bacteria in it, it would die from the boiling, but it would still be a good environment full of nutrition for other microbes to come in. So, in the flask that did not get broken or did not get tilt, there were no microbes formed at all. However, in the flasks that were either broken or tilted, there was microbial growth. This means that when exposed to air, dust, or other factors, life can and microbes can go into these areas, but only if they're exposed to outside factors. If there is no microbes, then they cannot grow whatsoever. Next, we're going to talk about the Miller-Urey experiment. But before we do that, we must discuss about how the first cell came to be. First prokaryotic cell, I must be specific about. So in the 1920s, there was a hypothesis for how life could have originated on Earth. This was done by Alexander Oparin and J.B.S. Haldane. So they came up that there must be 4 conditions in order for there to be life on Earth. These conditions must mean there must have simple organic molecules that must be formed from inorganic molecules, as I stated here. There must be larger organic molecules to be formed from the smaller molecules. The organisms that are produced must be reproduce, and the replication of nucleic acids must be possible, and there must be self-contained structures necessary for biochemical reactions, as they need a set of conditions like pH. Now, Miller-Urey came up with this experiment, as we can see here. Now, this basically aimed to recreate the conditions of the Earth where life would be seen to be inhospitable, where the Earth would be seen to be inhospitable. So the experiment goes, firstly, water is boiled, and this is where it would be. Water is boiled and evaporates and moves up here into this larger flask. This combines with different gases like water, methane, ammonia, hydrogen gases, and sparks here are fired between electrodes to simulate lightning. Then this gas will move down the cooling condenser and turn back into liquid, which will drip down into this trap here, where organic molecules produced in the reactions will also settle. After a week, they found that simple amino acids formed and complex oily hydrocarbons also formed. This meant it ticked the first condition to be met, which means that organic molecules, including amino acids, can form from inorganic compounds. and thus organic molecules could have formed on prebiotic Earth. Next, we're going to talk about endosymbiotic theory. Now, as we discussed before, the first prokaryotic cells most likely came from inorganic compounds, right? So, what about eukaryotic cells? Well, to begin with, some organelles of eukaryotic cells, mainly the mitochondria and the chloroplast, are said to have not originated at the same time of eukaryotic cells. Evidence for this suggests that when we look at mitochondria and chloroplast, both have a double membrane, right? Which is akin to prokaryotic cells and eukaryotic cells, but them being their own cell. Another characteristic they have is that they both have 70 S ribosomes, which is very important considering that eukaryotic cells, as discussed before, have 80 S ribosomes. And another piece of evidence suggests is that they divide by binary fission, which is relative to prokaryotic cells. They are this is a relatively new one, but they are susceptible to some antibiotics. Now, as you go in through the unit, you'll realize how these antibiotics will work against bacteriums, which are mostly prokaryotic cells. And since they have the metabolisms and all that. So when we're looking at chloroplasts and mitochondria, it is very important to realize that if they get affected by these antibiotics, not only will it be difficult to manage in terms of the medical field, but it also provides a piece of evidence that they were once their own prokaryotic cells. I'm just going to write here that relates sweet. Now, in addition, one more piece of evidence suggests, or states rather, they have circular DNA. It is not protein related, so there's no histones or whatnot. Now, these are all pieces of evidence for endosymbiotic theory, right? But the actual theory suggests that during some time when there was a lot of prokaryotic cells, not much life, right? There was a larger prokaryotic cell. Let's just say this is the prokaryote. And L will be for large. And there was a smaller one, which could be the mitochondria, chloroplast, or some relation to it. Let's just say S prokaryote. Now, at some point, since the main food source is through eating other bacteria, other prokaryotes through engulfing, this large prokaryote, I'll just refer to it as LP, and this is SP, make it easier. It would have began to engulf the smaller prokaryote and then eventually fully consume it. I'll just give it some color to differentiate. Now, at this point, instead of digesting the cell, or rather digesting the prokaryote, since the nature of mitochondria and chloroplasts are to provide energy for the cell, for the eukaryote, this prokaryote that engulfed it, since it was beneficial for it to keep it, it did not digest it, and rather it lived as a part of it. So to provide it energy for whatever. And that is essentially the symbiotic theory. Thank you for participating, and I hope you've all learned something from this.